Hello, everyone, and welcome to The Explainer. I'm your host, Dr. Chem. Today, we're diving into a true masterclass on one of the most powerful, highly selective separation techniques in modern biomedical science. If you've ever wondered how scientists isolate a single microscopic target from a chaotic biological soup with absolute precision, well, you're in exactly the right place. Today, we are mastering bioaffinity chromatography. Here's our syllabus for The Explainer today. First up, principles of recognition. Second, the anatomy of the column. Third, the step-by-step process. Fourth, the advantages and limitations. And finally, we'll wrap up with real-world applications. All right, let's jump straight into Section 1, Principles of Recognition, Finding the Needle. Now, imagine trying to find a specific needle in a haystack, but that haystack is also full of pins, nails, and wire. That's essentially the immense challenge of isolating a single target protein from a complex biological mixture. Bioaffinity chromatography is so uniquely powerful because it actually solves this by exploiting nature's own mechanisms. It relies entirely on biological recognition. We're talking about that highly specific, natural lock-and-key interaction between a target molecule and its binding partner. So whether that's an antigen binding to an antibody, an enzyme connecting with its substrate, or a receptor recognizing a ligand, we are essentially borrowing this flawless biological targeting system and putting it to work right there in the lab. Moving on to Section 2, Anatomy of the Column. Let's look inside the machine. So the real heart of this entire technique lives in the stationary phase, specifically in something called an immobilized ligand. This is a specific biological molecule, like an antibody, enzyme, or a nucleic acid, that is chemically coupled to an inert solid support matrix, something like agarose or silica. Think of these beads sitting inside the affinity column as millions of tiny, highly specific hands just waiting to catch their exact target partner. Now, there are a ton of different ligands we can use, but antibodies are incredibly common. Why? Because they offer massive specificity and very large binding constants. The crucial point here is that when we use monoclonal antibodies, which lack the variability of polyclonal ones, we create a highly uniform, reliable affinity support. It forms this basically unbreakable antibody-antigen interaction the exact moment the target flows by. But there's a catch. Sometimes the binding site of our target molecule is deeply buried. If we attach our ligand directly to the matrix, we actually get really inefficient binding because of steric hindrance. Basically, the molecules just physically bump into the matrix. But notice how adding a spacer arm, which is usually a chain of at least 3 atoms, pushes the ligand out. It gives it room to reach out and allows for deep, efficient binding. It's perfectly clear when you look at the classic structure of an antibody ligand, showcasing the specific Fab regions reaching out on those spacer arms to grab the target. All right, Section 3, the step-by-step process, how the system actually works. So how exactly do we turn these biological principles into a mechanical process? Well, the operation runs through a highly automated, high-performance liquid chromatography, or HPLC system. It starts at a solvent reservoir and degasser, flows through a high-pressure pump, and straight into an autosampler, where our mixture is injected. The sequence is super precise. First, we equilibrate the column with an application buffer. Second, we inject the sample. Third, the target binds. Fourth, we wash away the impurities. And finally, we elute, or release, our purified target right into a fraction collector. Now, step 3 is where the real magic happens, specific binding. While a tremendously complex biological sample flows through the column, those immobilized ligands literally ignore everything except their exact target. Our hero molecule is captured and held incredibly tightly, while all the unwanted cellular debris and impurity molecules simply flow straight past. It's a perfect molecular filter. This molecular cycle illustrates that perfectly. First, the complex sample is applied. You can think of it as a mix of circles, triangles, and squares. The Y-shaped antibodies selectively grab only the triangles. During the wash step, all those unwanted circles and squares are totally flushed out. Finally, in the elution step, the triangles are forced to detach, dropping down as a perfectly purified sample. And the best part? It leaves the column completely regenerated and ready for the very next run. And while all this is happening microscopically, our computerized data systems and detectors, like UV visible or LCMS, are tracking the entire process in real time. A typical chromatogram shows exactly what the computer is seeing. That first massive, broad bump, that's the sample application and the washing away of all that unbound material. But then, notice that sharp, distinct spike on the right? That peak represents our purified protein of interest, detaching and flowing past the detector in a highly concentrated form. To get that beautiful, sharp spike, we have to use an elution buffer. Because that biological interaction is so strong, once the impurities are gone, we actually have to chemically break that lock-in key bond. We do this by changing the pH, altering the ionic strength, or sometimes adding a competing ligand to the mobile phase. This essentially forces the target molecule to let go of the stationary phase so we can finally collect it. Which brings us to Section 4, Advantages and Limitations. Let's weigh the technique. Look, no analytical technique is absolutely perfect. Now, the advantages of bioaffinity chromatography are genuinely staggering. I mean, we're talking extremely high selectivity and literally thousands-fold purification efficiency in a single step, plus excellent recovery of biologically active molecules. But we have to impartially weigh the limitations too. The affinity ligands, especially the antibodies, carry a pretty high cost. There's also a risk of ligand leakage right into your purified sample. The ligands have limited stability, and methods like CNBR-activated Cepharos might only have a lifespan of about 15 cycles. Plus, the whole thing requires very carefully optimized binding and elution conditions to work properly. Finally, Section 5, Real-World Applications, the Global Health Impact. Despite those limitations, the raw power of this technique makes it universally utilized. So why does this matter to the world? Well, the scope of its applications is just massive. It's the absolute gold standard for protein purification, isolating enzymes, hormones, and therapeutic proteins for pharmaceutical research. It's absolutely crucial for biomarker isolation, letting us pull specific disease indicators right from blood or serum for clinical studies. And it's the foundation of diagnostic research, allowing us to prepare highly purified biomolecules for immunoassays, biosensors, drug discovery, and even large-scale vaccine manufacturing. And researchers are constantly tailoring these techniques for highly specific tasks. We don't just use biological ligands like staphylococcal protein A for capturing immunoglobulins anymore. We also engineer structural ligands. Think of the metal chelators used in immobilized metal ion affinity chromatography, or IMAC, which capture His-tagged recombinant proteins. We're even developing synthetic, biomimetic ligands that are highly resistant to degradation, which is opening up even more scalable purification options for the future. So, to summarize our masterclass today, bioaffinity chromatography is without a doubt completely indispensable in modern biomedical science. By simply exploiting the beautiful, natural, biological interactions that already exist in nature, this technique allows us to isolate target biomolecules with exceptional, totally unparalleled purity. Its outstanding specificity is exactly what makes modern biotechnology, proteomics, and pharmaceutical development possible today. I want to leave you with this provocative thought. As we continue to engineer increasingly precise synthetic and structural ligands, what completely new frontiers in personalized medicine and early disease detection will bioaffinity chromatography unlock next? It's a pretty amazing thing to consider. Thank you so much for attending this masterclass today. Keep questioning, keep exploring the microscopic world, and I'll see you in the next explainer.